everyone, my name is Giselle Sanchez. I am representing SACNAS Outreach at the University of Michigan. SACNAS stands for Society for the Advancement of Chicanos, Hispanics, and Native Americans for Science. I am also a graduate student at the University of Michigan. Being a graduate student means that I am pursuing higher education. In my case, I am pursuing a PhD or a doctorate's degree in pharmacology. The video you're about to watch features 4 different graduate students at the University of Michigan. These 4 students represent different fields of life and biological sciences. These scientists will give you a tour of their labs, as well as presenting the work that they do. I hope you enjoy.
Stephanie, and I'm originally from California. I'm from Concord, California, which is just east of Oakland and San Francisco. My parents are originally from El Salvador, and I am first-gen U.S. citizen and also college graduate. I first fell in love with the brain, which I'm lucky enough to study now, when I was in high school, and my teacher just taught us about the really basic parts about the brain. And it's really cool now because as a graduate student that studies neuroscience and how the brain functions and dysfunctions in disease, it's kind of cool to think back on those early high school years when I was first learning about the brain. And I'm in a lab that studies what happens in our neurons when our brains are sick, so specifically in diseases like Parkinson's disease and like Alzheimer's, when our neurons, our brain cells, can't really stay healthy anymore. And so something that I do pretty regularly is collect brain tissue from mice. And so here is a little half of a mouse brain that ultimately what I'll do is section it, take really, really thin sections from this guy and look at the neurons and look at different proteins in the brain, look at either how much of this protein there is or how it looks, if it's like clumping together like it does sometimes in disease. And so that's a pretty normal thing that I do on a regular basis in lab. So this is what our labs look like. It's really cool because all the benches are open, and so you can collaborate with other scientists and kind of just see what everyone's up to and ask for help when you need it. So today what I'm going to be doing is working with some human cells actually that we grow in a dish. And I've treated them with DNA so that they express the proteins that I want them to express at really high levels. And so now I'm going to use different drugs to inhibit different activity in the cell. To look at what's happening, and it's a really great way for us to understand how things work, is to either turn something on or to turn something off. And so I'm using kind of 2 different types of drugs, one that turns on certain pathways and another that turns off certain pathways, so then I can see any changes in the cells with those things. And so I'm just going to mix those up really quick so I have it all ready to go, so I can just put them on my cells in a second. Probably want to label things. Super important to label things in labs. Checking my cells to make sure that they look healthy and that they're doing well. I don't want to do experiments on unhealthy cells. This other microscope lets me look at them nice and close so I can see how they look.
Hola, hi there. My name is Loyda Morales and I'm a graduate student at the University of Michigan. In this video, I would like to share with you what I do in a science laboratory or lab and what I am studying as a graduate student. But before we get into that, I would like to share with you a little bit about me. So, I was born and raised in the mountainous area of Puerto Rico. The small island here within the red circle is Puerto Rico, a small island in the Caribbean near Haiti and the Dominican Republic. This other picture here shows the view I woke up to every morning. All of my K-12 schooling was in Puerto Rico at nearby public schools. I went to college at University of Puerto Rico, and I graduated with a bachelor's degree in biology in 2017 and became the first in my family to graduate college. After college, I worked for a year as a laboratory technician in a science lab. Here, I helped maintain the lab run smoothly. Here's a picture of me doing that. Here I am designing and performing experiments, and I also ordered lab supplies and materials as part of the job. These materials were then going to be used in experiments later on. After about 2 months of being a lab technician, 2 very powerful hurricanes, Category 5 hurricanes, hit many islands in the Caribbean. One of them was Puerto Rico. These 2 events left some of the research facilities, including the one I worked at, and many other homes and businesses without electricity, water, and internet for more than 4 months. But despite these 2 Category 5 hurricanes, I was able to do some research, gain techniques, and apply to graduate school to get a doctorate degree in pharmacology. Now, I am a graduate student at the University of Michigan here in Ann Arbor. And as a graduate student, I study human cells and how these cells use proteins to sense and respond to the outside. My research study will help scientists and doctors of tomorrow understand how these human cells are involved in fighting diseases like cancer. Because understanding how these acid-sensing proteins help fight human diseases will help create better therapies and treatments for patients suffering from cancer. This is the Putin B lab, where I work at. Here is the benchwork area, and here we do experiments that do not require living cells. And over here, come on with me, here is the tissue culture room, like the name says here. And here we take care of cells that we work with and maintain the cells alive for experiments later on. Like I mentioned before, I study human-like cells and how these cells use proteins to sense and respond to the outside. The proteins that I am studying are called acid-sensing proteins, and these help cells and the human body fight against diseases like cancer. Cancer happens when cells that are not normal grow and spread very fast. Cancer cells just don't die when they're supposed to, and create and maintain an acidic environment. The environment surrounding the cancer cells is acidic, and this acidic environment help cancer cells survive and spread cancer to other parts of the body. One of the proteins that cells use to sense and respond to acidic environments surrounding cancer cells are the acid-sensing proteins. These acid-sensing proteins help cells sense and respond to nearby acids and help fight against the spread of cancer and protect other cells from turning into cancer cells. But how do acid-sensing proteins help cells sense the acidic outside and respond? Well, that is the question that I'm trying to answer as a graduate student here at University of Michigan. To answer this question, I use human cells that have been isolated from people, expose these cells to an acidic outside liquid environment, and observe how the cells sense and respond to the outside using this right here, a microscope, which is a tool that helps make tiny objects, very, very tiny objects, appear very large. Here is one example of something we can do with a microscope. In this movie that I'm about to show you, you will see a cell, in fact, many cells, that contain a lot of acid-sensing proteins. Here, you can see, in fact, multiple cells that have the acid-sensing protein in white and are all around the cells and inside as well as little dots. By using a microscope and other tools, I'm able to visualize the acid-sensing proteins and see when it's turned on or when the protein is sensing the environment. I'm also able to see how the protein moves throughout the cell and where the protein goes, as well as how the protein's location within the cell affects the response of the normal cells to an acidic outside. Understanding how these acid-sensing proteins help fight human diseases will help create better therapies and treatments for patients suffering from cancer. Now, that is all I have for you. I hope you liked this video and learned a little bit about what I do as a scientist and what I study.
So hi, my name is Freddy Cisneros. I'm a first-generation Mexican-American student. I'm a graduate student at the University of Michigan in the Applied Physics program, where I do theoretical and So the path that I took to get to where I am was kind of, I think it was a bit of wanting to pursue a certain path, but at the time, I think there was a lot of barriers that I had to face. I wasn't able to get into the research groups that I wanted to, and so then I had to go through a different path and find another research topic that I was interested in. And I eventually got to the path that I am. Since my work is theoretical and computational, I have the luxury of, taking my lab with me. So I can just take a notebook with me, like in my backpack, and I'm able to go like wherever, like to a cafe, out to the park, to the beach, wherever, and work from there. I'll start my day, and then I'll stumble on a problem like right away. And so I'll have to ride my bike, or I'll have to go skateboarding, or I'll have to go take a walk. That'll be like 3 hours, 4 hours. And then I'll come back to it. And then maybe it'll work out, maybe it won't. And so I'll just spend maybe I'll try to work on another problem that I'm trying to understand, But so what gets me excited about my research is that the work that I do is relevant to biology. So and chemistry, a lot of the life sciences. So it's really awesome to be a physicist and be able to work with biologists and chemists, or do work that they can use like sometime in the future. Because like when I was younger, I wanted to be a doctor but things didn't work out, and eventually I decided to become a physicist. But often physicists get disconnected from from the world, like in reality. And so it's pretty cool to caring for people, because if the research that I do goes to biophysics, that could eventually let's say lead to the cure for cancer or something like that. That's really awesome. And so that makes me want to work harder. One of the projects that I'm currently working on has to do with studying active matter systems. Active matter systems are systems where the particles, they'll go in straight motions, or they'll periodically change their direction. So when these clusters form, they'll just aggregate more particles. More particles will collide into these clusters and form larger clusters. But often, particles would also leave these clusters. So you have, just the cluster that keeps changing size and shape. So often what you see, the motion that these particles have inside the balloon is similar to what's going on inside this piston. Is that they'll just go through random motion, and they won't interact with one another. So the physics of this type of gas, which is known as an ideal gas, it's well understood. We know the physics of it. The thing that we want to do with these active matter systems is understand the physics, because we don't quite understand. But this can be something that we can represent as cells aggregating inside the body. If cells that lead to blood clots can be represented as active matter, then it's very, very important that we understand active matter, so that we can understand how blood clots form, and so that we can reduce the formation of blood clots. And so I'm actually going to be working with the group in France as part of my Fulbright fellowship to understand the physics of these active matter systems. And so this is really interesting research because not like we don't, physicists don't really understand too much about this. So there's many questions. There's a lot to learn about these systems. So which makes it even more attractive.
Hi everyone, I'm Natalie Momplaisir, but you can also call me Nat. I'm a second year PhD student here at the University of Michigan. Before I tell you all about this science, let me tell you a little bit about myself. I originally came from the Caribbean island referred to as Haiti, and then I immigrated here in 2013, and I finished my high school and went to college and studied chemistry. Usually chemistry is hard, but it's actually pretty cool. So when I graduated with my bachelor's degree, I came here to the University of Michigan to study pharmacology. And then with pharmacology, I can actually do the chemistry, but really understand how all these medicines that you take work inside the body. Now let's say that grandpa or grandma goes to the doctor and they have a specific heart disease, and you see the doctor giving them all of these medicines. Do you ever sit down and think, how do these medicines work? Where do they go in the body? What do they do? Well, all I've studied a specific receptor that is all over the body called G protein-coupled receptors. And these medicine can actually target these receptors to help your grandpa or grandma feel better. Now that I have my lab coat on and my gloves on, let me show you the pretty cool stuff that our lab has. This is our chemical bench where we have all of our chemicals and our balances to weigh all these chemicals for our experiments. And then we also have pretty cool machines in our lab. And here's a pretty cool one. We have this machine that aspirates liquid from this container and drops it to this plate reader over there. Now that I have all my liquid dispensed here, we can use another cool machine in the lab that a lot of the scientists in the lab use a lot. It allows us to look at the fluorescence and absorbance of our compounds, but also in cells. But here's my favorite instrument in lab. That is the centrifuge. It allows us to spin samples, and I don't know what it does. Now that I've showed you all these cool machines that our lab has, let me tell you what I, as a scientist and a pharmacologist, do in the lab. So like I mentioned before, I'm interested in finding out and understanding which proteins interact with these GPCRs. And so to do that, again, I would use cells and put the DNA that encodes for the protein and then visualize that interaction. But first, how do we have that DNA available to us? We have bacteria that can help us replicate our DNA that encodes for our protein. And then, once we have all these bacterial colonies here, we can collect the DNA and then purify it so we can have it in our cells. Another place in the lab where I spend a lot of my time is this tissue culture room here. So this tissue culture room, that's where I take out all of my cells and keep them happy. But to take care of them, I need to place them in an incubator that keeps them very warm and very happy, and so I can use them for my experiments. So here I'm just going to be taking my cells out of the incubator. And we usually keep them in this dish so they can be happy and just spread and touch their friends. But again, taking care of them, I can't just leave them outside, okay? So I have to leave them in the sterile hood where I can give them food and use them for all of my experiments. And that's when I keep them away from all the germs in the outside world, and I can just work on them here. Now, after taking care of my cells, I want to see if they're happy or if they're sad. But I can't do that with my naked eye, and you can't either, unfortunately. But guess what? We have this instrument here, which is a microscope, that can help us visualize how happy our cells look today. So first, I'm going to go through these lenses and see how happy my cells are. They look pretty happy, but I want you to see how they look. So I'm going to turn on the camera that connects the microscope to the computer, and so you can see how happy the cells look today. Wow, come look how happy the cells are. Look how they're touching their neighbors. I actually did a very cool experiment for you today. Remember these bacterial colonies that I showed you earlier, and I said that I could take the DNA? I actually did, and I put it in my cells. Now this DNA encodes a green protein that will make my cells glow green. Isn't that just amazing that we can do that? So now I want to see how my cells turn green and how happy they are with that green color. So what I'm going to do is change my filter in the same microscope. And oh my gosh, these cells are pretty and glowing green. Oh my gosh. Happy cells, happy scientists. I'm so happy you took the time today to listen to me and my science talk today. And as you know, science rules always. And I can't wait to see the amazing scientists that you will all be one day.